So in the last video, we discussed memory, how memory is stored sort of on a global level, as well as the various anatomical structures of the brain that are responsible for mediating the storage of memory. I want to switch now to a smaller scale and talk about the molecular mechanisms, and I'm going to focus specifically on declarative memory because that's what people have studied. It's probably true that the same sorts of mechanisms, perhaps in different brain areas, but the similar mechanisms are involved in all non-declarative memories as well. So remember that we said memories are stored as distributed connections of between neurons within the cortex. But I want to, I guess, add that the memories are stored as segregated patterns of connections. And what I mean by that is that we said that memories are distributed, and this is one of the theories of how memories are distributed between the hippocampus and the cortex of the brain. So there are traces between the hippocampus and various neurons within the cortex, and those traces, when recalled, enter into our short-term memory again, and they need to be reconsolidated. And each time they're recalled and reconsolidated, because we're in slightly different settings during those recalls and reconsolidations, there are slightly different traces. So there are overlaps, but the traces are different. What we didn't say last time is that, so when we followed Hebb's theory, we said that the connections between the neurons were strengthened each time they were recalled. And that strengthening is true, and we'll talk about molecularly how that strengthening happens. But it's not only strengthening, it's also strengthening of some and weakening of others, and maintenance of some and not maintenance of others. So it's the pattern sort of how the connections are made that allows us to, I'll say, recognize a memory. So on the right-hand side of this slide, I've got an image from your textbook of a simple circuit where there are three different neurons, A, B, and C, and each of these three neurons has connections with three other, we'll say, sensory neurons. And we are going to use this simple circuit to recognize three different people, three different guys, one named Mark, one named Barry, and one named Mike. So before we know Mark, Barry, and Mike, and we meet them for the first time, we see their faces, we hear their voices, and we don't know who is Mark and who is Barry and who is Mike. So all three of those neurons, A, B, and C, are stimulated to an equivalent level. However, after we learn who is Mark, we see that neuron A has a strengthened stimulus when we see and or hear Mark, versus neuron C, which has a weakened stimulus when we see or hear Mark. Compare that to Mike. When we see or hear Mike, neuron A has a decreased stimulus, but neuron C has a wow over the top stimulus. And compare that with Barry in the middle. Barry's stimulus, or all three of the neurons, A, B, and C, have an enhanced stimulus relative to what it was before we knew who Barry was. But in some cases, that stimulus is greater than somebody else's. So for neuron A, for example, Barry's stimulus is greater than Mike's, but it is less than Mark's. So what I'm saying is that it's the pattern of which neurons receive an extraordinary stimulus, a strong stimulus, and which neurons receive a less than strong stimulus. And it's that pattern that helps us recognize who is Mark and who is Barry. Now, I'm reminding you that when we are making memories, sensory information comes into our cortex and then it moves from, so this might, our somatosensory cortex, for example, or our visual cortex. And then that information travels to the parahippocampal cortex, the entorhinal cortex, and then it moves from that entorhinal cortex to the hippocampus. I want to look specifically at the neuronal circuit that's involved, and we call it the trisynaptic circuit because there are 3 different neurons that are involved. The first of those neurons is in the entorhinal cortex, and so information coming from some other cortical area is going to enter this entorhinal cortex and synapse on a neuron that will move via what we call the perforant pathway to the hippocampus. So this is a pathway of neurons from the entorhinal cortex to the hippocampus. At the hippocampus, and specifically in the dentate gyrus of the hippocampus, there's going to be a synapse on a second neuron in this trisynaptic circuit, and this second neuron is called a CA3, and that CA stands for Amon's horn in an English version of the Latin, and so the CA3 neuron has its receptors, or it's receiving the signals in the dentate gyrus, and then it will move through the hippocampus, and it will actually branch in the hippocampus. One branch of that neuron will go to the fornix, and the second branch is called the Schaefer collateral, and that Schaefer collateral will synapse on the third neuron in the trisynaptic circuit, and that is called a CA1 neuron. So the CA1 neuron is sort of at the terminus of this trisynaptic circuit. We're going to look at the connection between the CA3 and the CA1 neurons, and we're going to look at the EPSPs that are generated, so the excitatory potentials that are generated from the CA3 or from stimulus of the CA3 of the CA1 neuron. So what we're going to do is so this actually, by the way, was accomplished, I'll say ex vivo. So somebody figured out how to pull out the hippocampus and the entorhinal cortex and slice it and get a very thin section. And in that thin section that could be kept alive ex vivo for a period of time, we could pinpoint or isolate the CA3 neurons and add an electric shock that traveled similarly to an action potential, such that it reached the axon terminus of the CA3 neuron, released neurotransmitter onto the CA1 neuron, and we can measure the excitatory potential by looking at the voltage inside and the voltage across the membrane of that CA1 neuron. So again, we're going to stimulate the CA3 neuron, and we've got 2 different CA3 neurons that we can use to compare against one another. So we've got this blue labeled CA3 neuron, and we also have a red labeled CA3 neuron, and we could stimulate with an electric shock either of those separately so that they can send a signal to the CA1 neuron, and we can measure the response of the CA1 neuron by looking at its voltage across the membrane as an EPSP. In the top graph, we're looking at the stimulus of the blue, we'll say, CA3 neuron, and in the bottom graph, we're looking at the stimulation of the red CA3 neuron. And in this case, that is simply used as a reference. So the red labeled input 2 CA3 neuron is a reference point for us. Both of these CA3 neurons are going to be stimulated at a rate of one stimulus, so one electric pulse every minute. So that's simply just providing us with a baseline or a background to basically tell us that the CA1 neuron is responsive and alive and all of that sort of stuff. So we're going to get this baseline response, and this baseline is every time we give an electric stimulus through the blue input 1 CA3 neuron, we're going to get some level of EPSP that we're going to say is 1, and that is our baseline. And then what we're going to do is we're going to pulse by providing 100 stimuli per second, boom, 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 like that. And then we're going to look at what is the voltage across the membrane as a result of that rapid response. And then we're going to return to this one stimulus per minute, and we're going to measure the EPSP every time one of these stimuli come in. And what we can see in the top graph is that following that very rapid stimulation, high frequency stimulation, that the baseline has shifted from our 1 to something greater than 1, right? So there is an increased stimulus every time, or an increased response every time a stimulus comes in. We call this long-term potentiation, or LTP. Long-term potentiation simply means that the CA1 cell is responding to a greater degree to the same amount of stimulus as a result of that rapid high frequency stimulus that came for just a short period of time. So the CA1 cell is, let's say, listening more astutely to the CA3 cell that it got that high frequency stimulation from. That this is long-term potentiation, and it is part of what Hebb described as a stronger connection between these two neurons. Long-term potentiation can be long-lasting, and so this diagram here, or this graph here, shows you the amplitude of the EPSP. So how strong is the EPSP above 100%? So 100% is this dotted line right here. So this tells us what baseline is, and we've got LTP greater than that 100% baseline as a result of some stimulation. And we see that it's lasting 360 days. So that's a year. Now, I'm going to talk about the different things that are needed in order to make an EPS, or to make this LTP last that long, because it's not necessarily automatic. Sorry, I'm finding my clicker. Okay, so in the previous slide, we were talking about doing this high frequency stimulation for a short period of time to produce long-term potentiation. And that's what's shown down here at the bottom as temporal summation. Hopefully you remember this as temporal summation, where you can produce a lot of action potentials over like repetitive, boom, 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 boom over time. Realize that there's also such a thing, or remember that there's also such a thing as spatial summation. So if the downstream CA1 cell received a bunch of stimuli from a number of different neurons, CA3 cells in space, or if it were from different collaterals of a CA3 cell, then you would have spatial summation. So long-term potentiation can result from either temporal summation, which was the example that we used, or spatial summation. We're talking about EPSPs, so these are excitatory potentials, and we're also talking about the central nervous system, so that should tell you that since glutamate is the most common neurotransmitter in the central nervous system, that the cells we're talking about, the CA3 cells and the CA1 cells, they're using glutamate as their neurotransmitter. What I'm looking at specifically is the synapse between the CA3 and the CA1. So the CA3 cells are going to be using glutamate, and that glutamate is going to bind to glutamate receptors, and we're looking at specifically the ionotropic receptors because those are the ones that were most commonly studied, but realize that there are also metabotropic glutamate receptors on the CA1 cells. So we're not going to talk about how those influence memory, but realize that they are there and they do have an impact. So there were two different types of ionotropic glutamate receptors that we spoke about, and those are the AMPA receptor that happens to be an ion channel for sodium, and the NMDA receptor that happens to be an ion channel for calcium. The NMDA receptor is, I'll say, has a little bit of a delayed response because in order for the NMDA receptor to respond to glutamate, there first has to be some sort of depolarizing stimulus such that the depolarization will allow for that magnesium ion to be removed from the blocking of the NMDA receptor. Once the magnesium is sort of kicked out of that blocking site, then the NMDA receptor can open and allow calcium in. But again, that takes some sort of prior depolarizing stimulus. So these two glutamate receptors are working in concert with one another on the CA1 cells. The calcium that enters into the CA1 cell is involved in LTP, and we know that because we can use chemicals to, and I put sort of in quotes here, suck up the calcium that moves through the NMDA receptor, so we can bind up that calcium so the calcium is not effective. And if we bind up that calcium and not let it run amok inside of the CA1 cell, we do not have long-term potentiation. So the NMDA receptor and the calcium that moves through it is critical for long-term potentiation. So how does calcium do that? Well, I need to remind you first of one way that we can modify proteins. So we're looking at a green globular protein on the left-hand side of this diagram, and this green globular protein happens to have as one of its amino acids a serine. The serine side chain is shown in pink, and what you can see is the serine side chain terminates in an OH group. That OH, oxygen and hydrogen, is a reactive group called a hydroxyl group that can be modified by the addition of a phosphate. And that phosphate is added enzymatically by a group of enzymes known as protein kinases. So protein kinases are enzymes that will transfer a phosphate group, from ATP onto proteins, specifically onto amino acids that terminate in an OH. Serine happens to be one of those. So on the right-hand side of this diagram, we see the same green globular protein that has this phosphate group transferred from ATP to the OH. The H goes away and we're left with this O linkage between the phosphate group and the serine side chain on what is now called the phosphorylated protein. So and I talk about this in a lot of my classes, and students will talk about proteins being phosphatized or phosphated, and the correct term is phosphorylated. So we have a phosphorylated protein, and what that means again is that a phosphate group has been transferred from ATP to an amino acid side chain by a protein kinase. In the case of LTP, long-term potentiation, proteins are going to be phosphorylated, and when they are phosphorylated, when this phosphate group is transferred to them, they change their shape, and that shape change will result in the protein being active. So the phosphorylated protein is an active protein. Once a protein is phosphorylated, it doesn't necessarily mean that it stays phosphorylated. This is a reversible reaction, and an enzyme known as a protein phosphatase can remove that phosphate group, returning the protein to its unphosphorylated form. And in this case, the unphosphorylated form is an inactive protein. So in this way, protein phosphorylation and dephosphorylation is a switch between turning a protein on and turning a protein off. So what does that have to do with calcium? Well, the calcium that enters into the CA1 cell is going to bind to two different protein kinases. We have seen calcium and calmodulin binding to proteins to allow a neurotransmitter containing vesicle to fuse with the terminal membrane at an axon terminus. We've seen that before when we were talking about synaptic transmission. Now the calcium that's going to enter into the CA1 cell is going to bind to either protein kinase C or CAM kinase 2, or CAMK2 it's sometimes called. CAM kinase 2 and protein kinase C are going to be two effector proteins that become activated when calcium binds to them. Since they are protein kinases, binding of calcium to these two proteins will allow for them to be turned on to become active kinase proteins, so that their enzymatic or catalytic domains, the active part of these two protein kinases, will become active, and that will result in their ability to phosphorylate proteins within the CA1 cell. So again, glutamate from a CA3 will bind to an AMPA receptor. The AMPA receptor will open to depolarize the CA1 cell. That will allow for magnesium to be kicked out of the NMDA receptor, and that allows calcium into the CA1 cell, and the calcium will bind to two protein kinases. Actually, it's going to bind to more than two protein kinases. We'll focus on these two right now, but that calcium will bind to two protein kinases, and that will activate these two protein kinases, who will go on and phosphorylate other proteins. One of the proteins that's going to be phosphorylated in the CA1 cell, and specifically that will be phosphorylated by protein kinase C that's been activated by calcium binding. One of the substrates of that, or one of the targets of protein kinase C, is the AMPA receptor. When the AMPA receptor is phosphorylated by protein kinase C, it's open for a longer period of time in response to glutamate. So if it's open for a longer period of time, even for a fraction of a second, which is what it is, more sodium will enter the CA1 cell and will have a greater depolarizing effect. Sodium will not have a greater depolarizing effect in general, but more sodium will have a greater depolarizing effect on the CA1 cell. So that will allow the magnesium from the NMDA receptor to be kicked out sooner, and that means that calcium is going to come in faster, so we're going to have a more robust response as a result of protein kinase C activation and phosphorylation of the AMPA receptor. In addition, CAMK2 will phosphorylate vesicles, or proteins in a vesicle that will allow for those vesicles to fuse with the postsynaptic membrane, and that membrane, or that vesicular fusion, will allow for the insertion of more AMPA receptors, a greater number of AMPA receptors in the CA1 membrane. So CAMK2 phosphorylates proteins on vesicles that leads to more AMPA receptors in the CA1 membrane. So in addition, the new AMPA receptors that are inserted into the membrane contain a specific subunit that has 2 phosphorylation sites, so it can be doubly phosphorylated and therefore stay open for an even longer period of time. We know that blocking of the NMDA receptors blocks learning, so this verifies to us that calcium moving in through the NMDA receptors is really what's responsible for long-term potentiation, and that is involved in memory. So it's not just looking at a series of molecular events that is like a gee whiz sort of thing. This is the mechanism by which learning occurs. Blocking CAM kinase activity also blocks learning, so this tells us that it's not only the NMDA receptors, but obviously the AMPA receptors that are upregulated, so we get more of those in response to CAM kinase 2 that is associated with learning and memory. And finally, if we increase the number of NMDA receptors, and we can do this through genetic manipulation of animals, if we increase the number of NMDA receptors, that increases learning capacity, meaning that the animals in which NMDA receptors are increased in number, those animals learn faster. So pretty cool. In addition to modifying the number of AMPA receptors and the phosphorylation status of AMPA receptors, long-term potentiation is also associated with changes in neuronal morphology, and so the shape of the neuron is going to change. So here are a series of pictures in which dendritic spines are glowing. So these are the white spots, or the white regions on the images in the middle of this slide. And we're looking at the one that's marked in the second to the left panel at time 0 with the yellow dot. That is the dendritic spine that we're going to focus upon. As soon as we add this stimulation to induce a very strong EPSP in a CA1 cell, we see 1 minute after that the dendritic spine has increased in size. And that increase in size is carried out for a full hour. So comparing the size of the dendritic spine before the stimulus and an hour after the stimulus, we see that that morphology change is maintained for that duration. So we're looking at a change in the size of the dendritic spines specifically, but it can also be the number of dendritic spines. But this diagram is showing us the size of dendritic spines is enhanced as a result of long-term potentiation. This is exactly what Hebb was talking about. He was talking about changes in neurons, both in morphology and in molecular composition. This is the strengthening of neuronal connections that he was talking about. So while Hebb was referring to long-term potentiation, the strengthening of these connections to help us maintain memories, I've told you that we also, because our memories are stored in patterns, we also have something that's called depression. So if we're going to compare what's happening in neuron A when we're looking at Mike, both before we know who Mike was and after we know who Mike is, we see that in neuron A, we have less of a stimulus after learning who Mike is. And similarly, we see the same thing with Mark, although in neuron C. So there is a decrease in the strength of that connection, and it's that pattern of increases and decreases in strength of neuronal connections that are stored as memories. So we have to have something that is depressing or diminishing that information or that stimulus. And so we have something called long-term depression. And we're going to use the same setup with the isolated ex vivo entorhinal cortex and hippocampus section, and we're going to stimulate two different CA3 neurons electrically so that there is glutamate released on a CA1 neuron, and we're going to measure the EPSP that's produced in that CA1 neuron as a result. So we're going to measure baseline again, and that baseline is going to be at 1 stimulus per minute. And then we're going to have a temporal summation, but that temporal summation is simply going to be 1 stimulus a second, which is 100 times slower than the rate of stimulus that induced long-term potentiation. We see after that, that we'll call it a weak stimulus, we'll return to that 1 stimulus per minute, but in response to that series of weak stimuli, or very infrequent stimuli, we have a less than 100% EPSP, less than baseline, and we call this long-term depression. So long-term depression is when we have that weakened stimulus, weakened connection as a result of a meh sort of input or meh sort of stimulus. Both long-term potentiation and long-term depression depend upon NMDA activation, but obviously of different levels of NMDA activation. On the left-hand side of this graph, we see long-term depression, and we see that long-term depression is associated with a small amount of NMDA activation. So this is a weak response, low levels of NMDA activation, that's associated with long-term depression. Compare that to the right side of the graph, where we have high levels of NMDA receptor activation, and that is associated with long-term potentiation. The way that that happens depends upon a ratio of protein phosphorylation. So looking at the diagram that's on the left, that's a synapse between, here it's labeled presynaptic and postsynaptic, but in our hippocampus, we're looking at a CA3 and a CA1 neuron. So those neurons will communicate, glutamate will be released, and that glutamate will bind to an AMPA receptor, not shown. The AMPA receptor will open, will depolarize the CA1 neuron sufficiently enough to open up the NMDA receptor, to remove that magnesium, to open up that NMDA receptor, and that will allow that calcium in, right? That calcium that will bind to our different protein kinases. The NMDA receptor will open, although not as many of them, so a few activated NMDA receptors will open in response to a low frequency stimulation. So that is the LFS, shown on the bottom of the right-hand part of this slide. High frequency stimulation, on the other hand, will allow for a lot more calcium to come in. So we're looking at the amounts of calcium that are coming in as a result of high frequency stimulation, a lot of calcium. So we have a concentration of calcium that is greater than 5 micromolar. And in low frequency stimulation, we have a concentration of calcium that's approximately 5 times less. When we have high frequency stimulation, we're going to have a lot of active protein kinases, and therefore we're going to have a greater amount of phosphorylated protein. And that greater amount of phosphorylated protein is going to yield long-term potentiation. When we have low-level stimulation, low-frequency stimulation, and therefore low levels of calcium, we're going to tip the balance such that we have more active protein phosphatases, and therefore we're going to have more unphosphorylated protein. More unphosphorylated protein is going to yield long-term depression. So what I'm telling you is that it's the ratio of phosphorylated and unphosphorylated protein, and specifically the phosphorylated and unphosphorylated AMPA, that is going to determine whether we potentiate or depress the CA1 cell. Now, that's not set in stone because, well, that it is what it is, but that new baseline is what we're going to call our new normal. And so we can potentiate or depress even after we have already potentiated or depressed. So your textbook shows a diagram of an egg carton, and I think that that's silly. This is not a ton better, but what I do want to show you is that this sliding scale sort of scenario where we can have a baseline, and because of infrequent stimuli, we can depress that connection and weaken a connection, but then that weak connection could potentially at some other point be strengthened. So our normal is just our normal right now. Your textbook describes this resetting of what the baseline is, this sliding scale of baseline, as metaplasticity of memory. So I also want to point out that the NMDA receptor, not all NMDA receptors are created equally. NMDA receptors, these ionotropic receptors, are always comprised of 4 different subunits that span the plasma membrane, the cell's membrane. And there are 2 of one type of subunit, so we're going to call it NMDA receptor 1 subunit or NR1 subunit, and those are identified in blue. And then we also have 2 of another type of subunits, these NMDA receptor 2 subunits or NR2 subunits. So 2 NR1s, 2 NR2s make up our NMDA receptor. you'll see that the yellow NR2 subunits in this diagram are labeled NR2A. Well, there are actually multiple forms of NR2, and another is NR2B. So some NMDA receptors are a combination of NR1, NR2A subunits, and other NMDA receptors are combinations of NR1, NR2A, and NR2B. Again, always 4 subunits, but sometimes we have more NR2Bs, sometimes we have more NR2As. When we have more NR2Bs, we are in a depressed state. So long-term potentiation is favored when NR2B is high. When NR2B is high, we favor long-term potentiation because more calcium is going to be allowed through an NMDA receptor that is comprised of NR2B as opposed to NR2A. You might have to replay this a few times to get that, but the next slide will sort of say the same thing over again, so hopefully it will make sense. So we're going to establish our new normal because of the phosphorylated AMPA receptors that are there, but also because of the NMDA receptors that are there. And the NMDA receptors are going to alter their responsiveness dependent upon their composition, the subunits that make them up. So after depression, and here it's labeled after deprivation, but let's say we're in a state of long-term long-term depression, LTD. So here we are, and we've got a lot more NR2B than we have NR2As in our NMDA receptors. Or a better way to say that is we have more NMDA receptors that have at least one NR2B subunit. Some of them may have 2 NR2B subunits, but there are more NMDA receptors that have NR2B subunits. We're in a state of depression. Now we're going to have some stimulation. That stimulation will favor potentiation because we're in a depressed state. So basically we're sitting there, we're long-term depressed, and we are waiting for input. When that input comes, we're going to say hallelujah, and we're going to become potentiated as a result. The converse is also true. If we are in a potentiated state, so we have this strong connection, we have then more NR2A than we have NR2B. But we are just waiting to be depressed. So long-term depression is favored when we have more NR2A, and that is because our cortical activity, we're high. And so we're potentiated. So we are just waiting for everyone to just shut the F up, right? So that is why LTD is favored when we have more NR2A. Okay, so we have looked at the molecular mechanisms by which connections are strengthened and weakened, and that has to do with the NMDA receptors letting in calcium, and that calcium binding to protein kinases, and those protein kinases now active phosphorylating things like the AMPA receptor. And we're going to change the phosphorylation ratio to produce LTP or LTD, and we're going to also change the composition of NMDA receptor subunits to allow them to be more or less responsive to to the glutamate that binds to them. So this tells us how we have LTP or LTD, which is strengthened or weakened connections, but we haven't really talked about how we stabilize these patterns so that we make memories permanent. So we're talking about LTD and LTP, and the graphs that you've seen up till now have mostly been on the time scales of minutes to maybe an hour, right? But we're talking about memories that are going to last a lifetime. So how do we do that? Well, one of the things that we do need in order to make our memories more permanent is to have CAM kinase 2 or CAMK2 be persistently active. So it has to be turned on all of the time. And the reason that that's true is that we need to have AMPA receptors embedded in our membranes over and over again, because those AMPA receptors are always turning over. The ones that are in the membrane are going to be taken in, and new ones are going to be put in their place. So there's this turnover of receptors. In addition, we need to have AMPA receptors continually phosphorylated. Phosphorylation, remember, is a reversible state, and so those phosphates can just fall off because that happens, or phosphatases can take them off, so we need to put them back on. So we need to have persistently active kinases. We're looking at CAM kinase 2 specifically, and you can see that there are two different parts of CAM kinase 2. One part is the regulatory part, and the other is the catalytic part. So in this state, the CAM kinase 2 is inactive because the regulatory part of the kinase is kind of shielding the enzymatic or catalytic part of it, and so the catalytic part cannot go on and phosphorylate things. So this is an inactive protein kinase. When the NMDA receptor opens and calcium enters the cell, that calcium will complex with calmodulin and bind to the regulatory portion of the CAMK2, and that causes a shape change such that now the catalytic part is free to access its substrates, proteins like AMPA, that will be phosphorylated as a result of this shape change. CAM kinase 2 can phosphorylate AMPA, but it also can phosphorylate itself. This is referred to as autophosphorylation. And when CAM kinase phosphorylates itself, there is a phosphate group that it puts a big bulky chemical group in the way such that the regulatory portion of it cannot refold to turn the protein off. So in this case, CAM kinase 2 stays persistently on. It turns itself on and it stays on so that it can go on and phosphorylate proteins like AMPA and itself to maintain this persistent activity. Persistent activity leads to persistent phosphorylation of AMPA, so that you are always in this LTP sort of state. So this is part of the extension of the permanence of long-term potentiation. So the permanence of memory. Well, this slide is focusing on and your textbook focuses on CAM kinase 2, other kinase enzymes also play a role in this permanence or persistence. A couple that your textbook mentions are PSD95, so this is on the postsynaptic density, that's what the PSD stands for, side of the CA1 cell, and this is a membrane-associated protein kinase. And then there's also protein kinase M zeta that regulates AMPA number, and it also regulates protein synthesis near the membrane. So these other two kinases, persistently active, are contributing to the permanence of membrane. We have to have a lot of active kinases going on if we're going to maintain memory. The other part of the story is protein synthesis. So in the last slide, I told you that protein kinase M zeta was associated with protein synthesis. And in that case, protein synthesis is occurring in the cell very quickly, and that is because there are mRNAs that are sort of lying in wait, waiting to be translated. So they've already been produced, but they're just waiting to be translated into proteins. And if that doesn't make sense to you, you're going to have to go back and look at sort of our molecular genetics thing that we were talking about, how we make proteins. Protein synthesis is going to be required for memory consolidation and the formation of permanent memories. What we see in this slide is over time, we see a weak stimulation near the left side of the graph, and that produces a long-term potentiation that after a couple of hours drifts back toward baseline. At hour 6, we're going to follow that with a very strong stimulation. And now we have long-term potentiation, greater EPSP magnitude in response, and that lasts for several hours as a result of the protein synthesis that is occurring immediately after this strong stimulation. This slide, we're looking at the same sort of setup that we've seen a couple of times before, where we've got some CA3 neurons connecting to CA1, and we're measuring the strength or the magnitude of the EPSP in the CA1 cell as a result of stimulation of the CA3 cell. So we'll stimulate the CA3 cell, and we're going to produce a long-term potentiation, and that long-term potentiation right here is going to start to wane after a couple of hours, right? Well, after an hour-ish, it starts to go back down. But what we're going to do is we're going to strongly stimulate the other input, the other CA3 cell. The other CA3 cell is synapsing on that the same CA1 cell that saw the first stimulus. But now, as a result of this strong stimulus of the CA3 cell, we're going to have LTP in the CA1 cell. But we're also going to maintain the signal, sort of, we're maintaining that LTP that came in from the first stimulus. So we're talking now about sort of spatial summation of LTP, but the protein synthesis that's occurring at the site of synapse with the red CA3 cells is going to drive protein synthesis at the site of input from the blue CA3 cell. So protein synthesis is occurring in the CA3 cell irrespective of who input the stimulus. Well, the previous slides were talking about synthesis of proteins from pre-made RNAs that are sort of just lying in the cytoplasm waiting to be translated. We know that some protein synthesis needs to be preceded by transcription, so we need to turn on transcription. And I'm going to recall a very old friend that we met at the start of the semester called Krebs. Remember, Krebs is a cyclic AMP responsive protein. You don't have to remember that, but Krebs is a protein that can bind to DNA, and when it binds to DNA, it regulates transcription. In fact, we have two different forms of the Krebs protein. The one that's shown in this image is Krebs2, and Krebs2 is what we're going to call a repressor of transcription. In the presence of Krebs2, this gene shown in this little cartoon is not going to be transcribed. However, when we have a strong EPSP, we can displace that Krebs2 from the DNA, and we can replace it with Krebs1. Krebs1 is known as an activator of transcription. Now, it's not only displacing Krebs2 and replacing it with Krebs1 that is essential for transcription. In addition, all of those protein kinases that we've been talking about are going to be responsible for phosphorylating Krebs as well. Once Krebs is phosphorylated, gene transcription is activated, so we can start producing RNAs, and those RNAs will find their way to the cytoplasm and produce new proteins. The new proteins that are produced are responsible for maintaining changes in neuronal morphology. And this is sort of analogous to that black and white picture that I showed you before, where we were looking at dendritic spines, but now we're looking specifically at new connections or new synapses. After a learning experience, you see in the second panel to the left that a new synapse has formed as a result of that experience. And that new synapse forming is preceded by gene transcription and protein synthesis. We see in the middle panel that with prolonged experience or prolonged stimulus, that LTP is strengthened even further. So the connection is very strong, again, because we're building that up with more proteins. We also see that we're going to start destroying an old synapse, so we're going to eliminate that old synapse because we really want to pay attention to the new one we've just made. That new synapse is waning over time without that stimulus, without that experience, but as soon as we repeat it, now we can build up to that same strength. All of this needs protein synthesis. So this is the diagram from your textbook explaining how this happens in people, but it's very similar to a diagram in one of the boxes in your chapter where researchers were looking at aplasia, so sea snails, and they were looking at a gill withdrawal response. And just looking specifically at the diagram, so when stimulated, protein kinase a different protein kinase than the ones that we've been talking about, but a protein kinase nonetheless is going to be activated as a result of stimulating this gill response. So we're going to produce protein kinase A, and in the right-hand image, we're going to see that as a result of protein kinase A, we're going to phosphorylate Krebs, and we are going to begin transcribing new genes. Those new genes, once transcribed, will produce new proteins, and we can see in that diagram that those new proteins have resulted in the production of a second connection with the motor neuron in this case. So it seems as though phosphorylation and protein synthesis is required for long-term memory in animals as well as in humans. So it's across species. Finally, just a couple more slides here. I would be remiss if we were talking about memory and I did not bring up the loss of memory in Alzheimer's disease specifically. So Alzheimer's disease is associated with a couple of, I'll say, anatomical or microscopic changes in neurons. The first of those is tau tangles. Tau is actually a intracellular cytoskeletal protein. It is associated with the microtubules, and the microtubules are responsible for moving things inside the neuron. So it's a sort of a moving sidewalk inside neurons. When an individual develops Alzheimer's disease, we see that these microtubules get all tangled up inside neurons, creating what we're calling tau tangles or neurofibrillary tangles. And these tangles interfere with neuron function and ultimately result in neuronal death, or at least are associated with neuronal death. Whether they cause it or not, we don't know. In addition to tau tangles, Alzheimer's is also associated with beta amyloid plaques that are extracellular, so outside of the cells in the space. You don't have to know necessarily the mechanism by which these amyloid plaques are formed, but suffice it to say that there is a membrane protein called amyloid precursor protein that is cleaved a couple of times to produce extracellular beta amyloid, and extracellular beta amyloids will glom up together with other beta amyloids and produce these extracellular plaques. Both tau tangles and amyloid plaques are associated with Alzheimer's. We now think because drugs were put into clinical trials to prevent the production of amyloid plaques, and it was hypothesized that if we can prevent the amyloid plaques from forming, we can prevent the memory loss. Well, it turns out that that's actually not the case. Some people believe that perhaps we simply didn't give the drug soon enough in the progression of the Alzheimer's disease. Other people believe that perhaps the amyloid plaques are simply a result of the neuronal death, so they're filling in spaces as a result of the neuronal death and really have nothing to do with causing the neuronal death that is associated with Alzheimer's dementia. So bringing this all home, bringing this back to Hebb. Remember, Hebb says that memories are stored in pattern or in connections, and we've extended that to talk about patterns of LTP and LTD and depressed responses and enhanced responses between an engram of neurons in the cortex of the brain. So what happens in Alzheimer's is when those cells begin to die, we start losing some of the cells that are part of that engram, part of those connections. And over time, as more and more cells die, fewer cells are available to be called upon to help us recognize this as a circle. And in fact, now when most of the cells are gone, what the heck is it that we're looking at? And eventually that memory is gone because all of the cells or most of the cells that were involved in that pattern of connections have been lost. So that's the molecular basis of memory. I hope that all makes sense, and we will talk to you soon.